Hello everyone, welcome back to my channel once again. I would like to thank you all for your continuous support and motivation since my first video, and I came back here with another informative video, and in today's video I am going to talk about gene cloning. I'm going to talk about its type, bit of procedure, and I will be focusing mainly on gene cloning vectors, type of gene cloning vectors. So before going to the procedure, types and gene cloning vectors, if you do not know about what is gene cloning, you can find it on my previous video. I'm going to leave the link of my previous video in the description section so you can directly access that video from there and you can get information about what is gene cloning first. So before going to the content, if you like my video, please press subscribe button and press the bell icon to get notification first. It gives me a big motivation to continue this work. Now, I would like to start from gene cloning procedure. There are two types of gene cloning procedure. One use restriction enzyme and another do not use restriction enzyme. And using restriction enzyme, we follow the recombinant DNA technology. And if you do not know about the recombinant DNA technology and you do not have the background about recombinant DNA technology, I have made some videos before, so you can find the link from the description section and you can access the video and build some background information and some knowledge from these videos. So I'm not touching the detail about the recombinant DNA technology here, but I'm going to mention how gene cloning proceed using the restriction enzyme. Using the restriction enzyme, the desired fragment of DNA would be extracted by using restriction enzyme. We caught in certain DNA sequence in donor vector and we extracted desired fragment of the DNA, and in similar way the donor vector DNA would be caught using the restriction enzyme. So we will be then the donor fragment of DNA, or the fragment of DNA that we want to clone will be incorporated or will be recombined into the donor vector DNA. Then we transform this recombination into the specific host cell and we will grow them in specific media, and we will be extracting these cells and we will be extracting DNA for the further study. This way, using the restriction enzyme, we can proceed the gene cloning. The specific fragment of DNA that encodes for certain gene that we want to clone, we have to cut it out from the donor organism or donor cell or donor DNA, then into the specific vector DNA we have to recombine them, and this recombination will have to transform into the specific host cell and have to grow. This is the simple way how gene cloning proceed with using the restriction enzyme. And next one is gene cloning without using restriction enzyme. You know, restriction enzymes have limitations, certain limitations, such as sometimes we may not find the restriction sites in certain vector, but or certain DNA sequence, but we have to open up that vector or we have to clone certain fragment of DNA that encodes certain gene. And in this case, if you do not have the specific restriction enzyme, or sometime for example the restriction enzyme have restriction sites, but it cuts the longer fragment of DNA than we need or we require. What can we do in this case is we can do cloning procedure without using restriction enzyme. Because of easy way of doing cloning, nowadays the cloning procedure without using restriction enzyme is becoming more popular, and I am here giving you an example of Gibson assembly cloning which doesn't require any restriction enzyme for cloning purpose. And this cloning procedure works into different phases. One is BP reaction and another one is LR reaction. BP reaction use BP clonase and LR reaction use LR clonase as an enzyme. So during the BP clonase, what happened is the desired fragment of DNA that encodes a specific gene that we want to clone. What we do, we use ATTB and we do several cycle of PCR, then we will get ATTB flanked PCR product of our specific gene of interest. Then we should have a donor vector that has ATTP sites and that contains CCDB, and once we do the reaction using BP clonase, it will give us entry clone with our specific gene of interest having ATTL sites. So yeah, this entry clone having ATTL sites will be like recombined with the destination vector having ATTR site and having CCDB, and once we do the LR reaction using LR clonase, it will express our gene of interest in that destination vector that would be our clone. Mean in this way our gene of interest would be expressed into the destination vector and our gene of interest will present into that destination vector. In this way we do cloning without using any restriction enzyme. It is just look like a transformation method from from the donor DNA that we want to clone, we will transform into the donor vector, then we will get entry clone, then that entry clone in will transfer our gene of interest to the destination vector, and finally it will express into the into the destination vector. In this way we do the BP and LR reaction separately and we will get our desired clone. After we will have our clone into the destination vector, what we have to do, we have to transform them into the specific cell, and we can grow into the specific media, and we can extract and we can get our specific gene of interest later on. I have mentioned about the types of gene cloning that use restriction enzyme and that doesn't use restriction enzyme. Now it's time to know what are gene cloning vectors. In molecular biology, vector represent as a vehicle to artificially carry foreign genetic material into another cell. So these vehicles or these carriers are small pieces of DNA and can be stably maintained in an organism. So they can be either bacteria or virus or plasmid or bacterium. They are highly convenient for insertion and removal, and even after insertion and removal, they can be stably maintained in an organism. So once we add some insertion into the vector, they can easily can carry this insertion or foreign material of DNA and can transfer into the another organism. This is the definition of vectors in molecular biology. Now, I would like to move on to the types of cloning vectors. In molecular biology, we can use several types of cloning vectors based on the cloning types. So, usually we use plasmid, we use bacterial artificial chromosome, bacteriophage, yeast artificial chromosome, expression vectors, transcription vectors, and human artificial chromosome as well. Now, I would like to go with examples for each type of cloning vector. First is plasmid. Plasmid is small circular DNA in bacteria or in protozoa, and we use this circular DNA for manipulation or insertion of our gene of interest to clone into the specific cell. Examples presented here are PUC19 and PRB322 vectors, both are obtained from E. coli. The PUC19 vector is 2686 base pair long and PRB322 vector is 4361 base pair long. And both of these plasmid vectors have a specific recognition site for different restriction enzymes. So we can use the these restriction enzymes to cut the specific portion of these vectors, and we can insert our gene of interest and we can recombine with these vectors and we can transfer transfer into the specific cell, and we can clone our gene of interest. Next example is bacteriophage. Bacteriophage is a type of virus that can infect bacteria. So, because it can infect bacteria, the bacteriophage is called as bacteria eater as well. Because bacteriophage can infect bacteria, we can use this ability to introduce our targeted DNA or gene of interest into the bacteria using a virus. Using bacteriophage as a vector, what we can do is we can manipulate the viral genome or viral DNA, and we can introduce our gene of interest or targeted DNA to be cloned. So, our targeted DNA or gene of interest can be inserted into the viral genome or DNA, and we will get recombination. After the recombination, we have to transform this recombination into the viral cell. So, viral cell can infect the bacteria. So, by choosing the specific type of bacteria that can infect by virus, a particular type of virus where we have done the recombination, we can introduce our recombination into the bacterial cell, because once a virus infect the bacteria, it will release its nucleic acid. So once it releases its nucleic acid, it will release the viral DNA expressing our gene of interest into the bacteria. So once it infects the bacteria, it will release our recombination, and our recombination will clone within the bacterial cell. So we can use bacteriophage as a carrier or as a vector to transform our recombination into the bacterial cell, and we can get our clone within the bacterial cell. So we can extract from the bacteria and we can use it. Another example is bacterial artificial chromosome. Bacterial artificial chromosomes, in short form known as BAC, and these BACs are engineered DNA molecules and usually used to clone DNA sequences in bacterial cells, such as in E. coli. These BACs are often used in connection with DNA sequencing. Very large piece of organism's DNA segment can be inserted into these BSEs. The BSEs are just artificially created chromosome to use sequencing in bacterial cell. So using the bacterial artificial chromosome as a vector, we introduce our gene of interest to be cloned within bacterial artificial chromosome, and we have to recombine them. Once we have the recombination, now we have to introduce this recombination into the specific bacterial cell, and then the recombination having our gene of interest or targeted DNA will be cloned within the bacterial cell. Here I have presented an example of artificial bacterial chromosome from E. coli, and the transformation have done in the Escherichia coli or E. coli cell. Another vector example is now yeast artificial chromosome. Yeast artificial chromosomes are genetically engineered chromosomes. They are derived from the DNA of the yeast. Usually, we use the DNA from Saccharomyces cerevisiae, which is the model yeast, and then we have to ligate this engineered chromosome into the bacterial plasmid, and in this engineered chromosome, artificial yeast chromosome, we can insert a very large fragment of DNA, and we can clone this DNA into the yeast artificial chromosome, or we can physically map the sequencing as well. And this vector is not commonly in practice nowadays, but it has very good historic value. I am presenting here an example of yeast artificial chromosome, which has multiple sites and two different markers and two restriction recognition site for ECOR1 and BAMHI as well. So in this YSA vector, once we open up the vector using the ECOR1 restriction enzyme, it will develop open section having the sticky ends before marker A and the CEN. So we can insert our gene or DNA of interest there, and we can clone within specific bacterial cell. So East artificial chromosome is one of the example for cloning vectors for gene. However, it's not commonly using these days, but we do have a very good historic value for this kind of vectors. Here we have another type of example for gene cloning vectors, and this is expression vector. And expression vectors are specifically designed to express the transgene into the targeted cells. And generally, they have the promoter sequence that derives expression of the transgene. And the expression vectors are also known as expression construct, which is usually either of bacterial plasmid or virus, which is designed for the gene expression into the cells, either into the bacterial cells or into the viral cells. And this expression vector used to introduce a specific gene into a target cell so that we can clone our gene of interest. However, in the molecular biotechnology, the expression vectors are the basic tool for the production of protein. So once we express our desired gene of interest into the expression vector, then we introduce it into a specific cell, and it can commandeer then the cell's mechanism for the protein synthesis to produce the protein encoded by that gene. So usually expression vectors used to produce the protein encoded by the targeted gene. So here we can see the example of PZEX3X expression vector having protein binding site. So once we introduce the specific gene of interest that encodes certain protein into the expression vector, it will then induce to produce like a protein from that specific gene sequence. And we can have the different kind of expression vector for the bacteria, plant, yeast, or even for mammal as well. So in plant or in agriculture, we use the agrobacterium as a expression vector that express the certain type of protein that we are desiring for or that we want to be expressed. So in this way, we use expression vectors to express the specific gene of interest that encode specific protein. And if you want to clone this protein for further use or for the study or research use, we use expression vectors to express these gene or DNA fragment that encode certain specific proteins. Another example we have is a transcription vector. Transcription vectors are simpler vectors, but they are only capable of being transcribed but cannot be translated. So they can be replicated in a targeted cell but cannot expressed. So transcription vectors are just opposite of expression vectors because transcription vectors cannot express the specific fragment of DNA or gene recombined into the transcription vector. They just can replicate within the cell but cannot be expressed. So they are just likely opposite with the expression vectors because expression vectors do express the protein encoded by the specific DNA or gene, but the transcription vector, it cannot express the specific DNA or gene, but it only replicate. And Finally, we have an example of gene cloning vector is human artificial chromosome, which is called as HSC, and this HSC is a microchromosome which can act as a new chromosome in a population of human cells. And there are two different ways to create human artificial chromosome. First is to create a small microchromosome by altering a natural human chromosome, or by method involve the literal creation of a novel chromosome de novo. In this way, we can create human artificial chromosome. So you might be thinking, why human artificial chromosome? Human artificial chromosome have ability to stably contain extremely large genomic fragments, mean we can introduce large fragment of gene or DNA of our interest to be cloned into the HSC. Usually in recombinant DNA tech and other vectors we use in gene cloning, they do have ability to express a small fragment of DNA or gene, but HSC commonly used because it can contain it can accept to introduce large fragment of genomic material. Therefore, HSCs have been used to create transgenic animal, and HSCs have been used to treat human disease and for the production of some therapeutic products. So HSC also act as a recombination agent, mean we can introduce the desired fragment of DNA or gene into the human artificial chromosome and can clone our gene of interest into the human cell. So in this way, scientists have got success into the development of transgenic animal for the agricultural purpose, and got success to cure some diseases, and got success to develop some therapeutic products. One of the example of human artificial chromosome is 21HSC. And this was for today's session. I have explained about what are the types of gene cloning, how gene cloning proceeds, and what type of vectors we use in gene cloning. I hope you enjoyed this video. I hope you have got some understanding about the gene cloning and the vectors involved in gene cloning. And if you know something more examples about the gene cloning vectors, do not forget to mention in the comment section. We can have the discussion there as well. As well as, if you want me to cover certain aspects of the agricultural biotechnology or biotechnology in my future video, you also can let me know on the comment section. Thank you for keep watching my video till end. Please guys, do not forget to like, share and subscribe my channel. It gives me a very big motivation to continue this work. We will see you in next video. Till then, have a very good day.